Okay, so I think we should start this morning session. So good morning, everybody. So it's a great pleasure to introduce Jan Rozman, who is, as you know, is a great collaborator of IMPC, was leading with Nadine the working group on cardiometabolisms, and he has made a strong contribution to the analysis of the metabolisms phenotypes in the IMPC pipelines. So Jan, welcome. So Jan moved recently to the University of Luxembourg, and he's now going to introduce us about the platforms which are located here. So Jan, it's a real pleasure to have you today online to present us and introduce us your new platforms. Thank you so much.
Yes, good morning, everybody, and thank you very much, Jan, for the kind introduction. It's really good to be here, and I'm really sorry that I can't join you in Cape Town and being there in person, but that was not possible this time. Still, I think it may be good to kind of give this presentation, to give everybody a chance to learn about what I'm doing now and to maybe identify future collaboration. So I want to talk about bridging innovation and the impact of LCSB platforms on biomedical research. And I hope you hear and see everything. Okay. So the Luxembourg Center for Systems Biomedicine, LCSB, is one of the 4 interdisciplinary centers of the University of Luxembourg. This is a special thing of the UL. They have a strong focus on, or the university and the faculties have a strong focus on teaching, of course, and academic training. But in addition to that, there are 4 interdisciplinary centers focusing on really special areas. And the LCSB is a biomedical center focusing on neurodegeneration. Shortly in numbers, we have 270 staff members and 18 research groups or PI groups, so led by an individual principal investigator. And they're coming from 3 different areas, I would say. One is the classical research, clinical research groups, so clinicians, excuse me, clinicians working with humans at the bedside and investigating neurodegeneration like Alzheimer's disease, like Parkinson, and also others with the people. Then we have a strong group of fundamental researchers or research groups that are really working with animal models, for example, or different model systems to, at the end,
Yeah, sorry to interrupt you, but you are not in the presentation mode. You are now.
Okay.
You should move from one display to the other, I think. The display setting is not correct.
Yeah, wait. On my computer, it looks all right, but maybe I stop sharing for a second. Okay. That you tell me now and not at the end of the presentation.
I thought it was just for a few minutes, but it did
No, it's... Oh, nothing's happening.
No, we are still on the PowerPoint mode here.
Mm-hmm.
If it does it come from us or from the?
No, everything is frozen now on my side. Wait.
We can see your mouse moving, but So maybe you should stop sharing and reshare again. Maybe it will reset up the system. I don't know.
Yeah, apologies for that. That's really a pity. Now start again.
This is a miracle of technologies.
Yeah. Okay, wait.
You know, it's a human-based technology anyway.
Yeah.
I don't think you can afford something which will be better than human-made than human, but maybe misleading. But... So, can you share again your screen, or did you?
I try.
Okay, go to the presentation mode and slideshow maybe, and see what happens.
Okay, now someone is requesting remote access. Let's see.
So I think you should remove the use presenter view, for example. I think that's would be helpful.
Okay, someone is controlling my computer now.
Wow, my God. From South Africa. I guess. I hope so too.
Okay, so do you see the presentation mode now? Because I see it.
Unfortunately not. We are still on the PowerPoint mode, not the presenting mode, not the presenter mode, but the PowerPoint. Maybe you have two screens and I don't know.
That's really strange.
Yeah. Otherwise, maybe if you close your left window. Yes, I think we are going to use the PowerPoint mode. So you can just maybe remove the window where you have all these slides shown so that you get it to the whole screen, and that's will fit.
Okay, so
Should go on.
maybe I leave the Zoom because it's really
I think
yes,
remove this. Yeah, yes. But nothing happened when you click on the slideshow.
Oh my God. I don't understand what's going on, so maybe I leave and come back in a second and log in again. That's really...
Yes, maybe try this.
Yeah, I'm really sorry.
Yeah. Oh, no. Okay, yep. So let's try again, Jan.
Yep.
Hi, Jan.
Okay, so now I'm sharing my screen, and now I go to presentation mode.
Yes. The slideshow doesn't change. Nothing happened. So maybe we'll continue on that.
Okay, I try to. So do you see the second slide now, Luxembourg Center for Systems Biomedicine? Okay, we tried like this. I don't have too much animation or stuff in there for the sake of time. Well, anyway, so I talked about the 3 pillars of the Institute: clinical research, data science, and fundamental research, and altogether 18 PI groups. So we have something like 6 to 7 people working or coming from these different areas. The funding director actually is Rudi Balling. Most of you, or many of you, will know him. They started working in 2009, and the current director is Michael Henniker. And he's kind of reforming and renovating the LCSB according to what is needed in the field. So as I said, tackling neurodegeneration, this is what we are doing. So it's not anymore metabolism or diabetes and obesity I'm interested in, even though we have many, let's say, links between metabolic dysfunction and neurodegeneration, like vegan, high-fat diet, obesity, diabetes. These are all important prerequisites for getting the disease. And the problem is, and we are all aware of that, is despite tremendous progress in medicine in the last decades, doctors are still unable to treat many brain diseases at their course. And why is that? We have not yet understood in detail how these diseases arise, particularly the earliest changes in the brain happen decades before any clinical symptoms appear. And the structure now again of the LCSB, here on the left-hand side we have management, and here the PIs. You see small pictures of them. And in addition to that, from the very beginning, the LCSB introduced Center Scientific Services. These are platforms that offer state-of-the-art technology methods to support researchers in their work. And a quick question again, do you see only the slide or also the other stuff that is on the screen?
We can see the slides and also the slides which are on the side. Yeah.
Okay, good. Platforms, this is kind of small form of a research infrastructure. We are all working in research infrastructures like IMPC, like InfraFrontier, or heading facilities that are part of a research infrastructure. But also on the small level, a core facility is in research infrastructure that is there to advance biomedical research. And there has been many papers actually, but also a recent one where they pointed out and showed how science is still disconnected. The translational gap between basic science, clinical trials, and patient care in Alzheimer's disease, and how this is holding up the progress in understanding and treating the research. Also nicely shown in this picture. The revolutionary and transformative discoveries in modern biomedicine depend on state-of-the-art experimental setups and methodology. And of course, they can't be offered to everybody, but they need to be concentrated in research infrastructures or core facilities and then be used by researchers. And thereby cutting edge and economically efficient scientific methods can be used. What we have at the LCSB are 7 platforms. These resources are easily available to all research groups in Luxembourg and beyond. So it's mainly the university and LCSB, but we are also open to collaborate with people from other Luxembourgish entities and even industry. It's fee-based. This is very important. And we have customized services that are offered to national, international, on the national and international level. The portfolio, and it will describe that in more detail later on, contains state-of-the-art equipment in the fields of sequencing, single-cell analysis, metabolomics, animal research, bioinformatics, bioimaging, and high-throughput screening. So that's how I will talk about that later on. Good. The first platform is the sequencing or genomics platform, and every platform has a platform manager, so a really seasoned, dedicated researcher that is beyond the postdoc level and is capable of heading these units. And Rashi has really a platform, and that's very similar for the other platforms in a way. She has a real machine park of different methods that are important for sequencing samples. I won't go here into too much of a detail and jump to the next slide, because that shows nicely that the samples, the nature of samples, can be very different. It's disease research, it's microbiome, actually in metagenomics, environmental research. The samples come from humans, animals, and plants. And she's offering here a couple of different methods, shotgun, for example, transcriptomics, amplicon sequencing, targeted sequencing. So many different kinds of samples. And I just show you this figure here, or this slide, because it nicely shows the origin of the samples between 2017 and 2022. We had a total of more than 12,000 samples, and were RNA and DNA. Here we have different user groups, actually from LCSB, DLSM, this is a department here at the university, DIST and LRH, these are two entities or research institutions here in Luxembourg, and then here the different methods. This is not to impress you, but only to show that project management is key to optimize processes, really from starting the experiment or the project, doing the project, and then at the end reporting. So this is really defined and really important pipeline that we try to maintain all the time. And this is very similar for all platforms. Let me go to the second platform. This is the metabolomics platform headed by Christian Jäger. It's also, I would say, classical metabolomics platform. And I know some of you have similar ones. It's really different mass spectrometric methods are used. So, again, a kind of machine park is available. We have tailored workflows with these MS technologies, but we also offer user trainings and something that is special, I guess, and which is also sometimes a bit complicated, because the flexible and cost-effective instrument renting option is also one way how people can use the infrastructure, and that requires a lot of training and a lot of work maintaining this machine park. And here a wide range of different instruments can be offered, and again, we lost a lot of time and I won't go into detail, but what we plan to do is introducing proteomics and spatial metabolomics. And I guess there could be interesting links in IMPC where people, institutions that are member in IMPC can maybe offer interesting options for us to learn more in this field. Okay, the third platform is bioimaging, also very classical, I would say. It's headed by Paul Anthony, and it's really spanning between FACS, live cell imaging, and laser microdissection, a couple of instruments again. Paul is offering consulting, of course, and training, then different things regarding image analysis, data management, and computer vision. And here, I would say a really a big innovation is the multiphoton microscope that we have just set up and that is running now and generating images or producing images of the brain of single neurons and during actually behavioral tasks like running wheel or even in the open home cage. So very innovative, very cost intensive at the end, of course. And Paul's platform is also an example for how important project management is. So booking these instruments, people are using then the microscopes and generate their data. One really fascinating platform for me is the disease modeling and screening platform. We don't have a platform manager here at the moment. It's a joint platform actually between LCSB or the university and the Luxembourg Institute of Health, LIH. The platform is so cost intensive that we share the innovation, the investments here in the platform. And the idea is actually that we use cellular models for early drug discovery. So this is actually the traditional use of the platform, but we are now thinking about doing the next step, like this CRISPR screening. So what is happening is that we have a disease model. These can be patient-based cell lines, for example, that are kind of developed or expanded to the dimension of this platform. And here we have, you see, it's a kind of drawing because a photo of the room looks like just too messy. It's a small room, and all this equipment is squeezed into the room with robots, with a lot of automation. And what is happening then when the cells are dispensed on a multi-well plate, we administer drugs, sometimes drugs that we buy in from a library, and then look at different phenotypes at the end, phenotypes of these cell models. It can go up to organoids actually, and we observe then either metabolic readout or morphological readout that indicates there's an effect of the drug on the phenotype of the cell line. And the goal of the DMSP is to facilitate the identification of disease vulnerabilities, so changes or effects on the phenotype, unveil target mechanisms or identify drug candidates for the translation of findings into personalized medicine. So we really run projects where after 2 or 3 different prior project steps, I would say, like setting up the model itself, so making it really work for the pipeline, then administering the drugs in the pre-screen, and then finally really using patient cells that are expanded and where we test then the effect of different drugs on these cell lines. And this is beginning with a screen, and then we have at the end even a preclinical candidate that may be interesting for industry or patient organizations. And what we are planning right now is doing a similar thing like IMPC using, we want to use CRISPR, a CRISPR screen, a genome-wide CRISPR screen to knock out genes that it could be an informed strategy, but it could be also completely unbiased, that we knock out single genes and check what is the effect on the phenotype of the disease model, and even investigate the interaction with different compounds. So we're entering a field there which I find very interesting because it's not that much dependent on animal work, but my experience so far is that it's nothing like controlled, or it's of course controlled, but we have to deal with a lot of variability because these cells are also living organisms and they are affected by different things in the handling, temperature, for example, pH, etc. So everything needs to be highly controlled. And I would say personally that it's very good for screening, but it's still pretty far away for really replacing animal experiments. And with that, we come to the next platform. That's an aquatic platform with zebrafish that is headed by Maria Lorena Cordero. And that's also actually new to me and very fascinating to learn about zebrafish as an alternative animal model. She is offering all the different, let's say, phases or support during all the phases in a classical research project. So housing and husbandry support, also support regarding writing up ethic approvals and planning the experimental design. There's also a lot regarding equipment and technologies that they can offer that starts with microinjections for gene editing, but also automated behavioral observation of the animals. And that can be used for drug screening and customized services. And last but not least, training is also very important for the platform. So she's involved in many, many trainings that are offered by the universities to users. And then we get to something you're very familiar with, the rodent platform that's headed by Jalil Kovar. And I don't have to tell you too much about a mouse facility. We have something like 2,600 cages capacity with SOPF breeding, where a germ-free unit is also there, and we can do embryonic transfer and IVFs. What Jalil is offering in a similar way as Lorena, full support during all phases of the experiment. It's really like planning the experiment, writing up an animal welfare or an animal ethics application to really conduct the experiment, and up to then having the animals and collecting samples or whatever. And what is important, and I think it's really worth to stress it. Contrary to many other countries, here in Luxembourg, we have an increase in animal numbers, and especially in the mouse field. And this reflects the need of the university, of institutions like the LCSB, to use mice for biomedical research. Of course, it's not, we have the same legal regulations in place. So the different committees that evaluate the animal ethics applications, but at the end we have also, as we are still recruiting new PIs and junior groups, there is still a heavy use of animals, and that's is in kind of, well, I predict a kind of conflict during the next few years as politicians and animal welfare activists may have a problem with that. And with the animal facility, I see, of course, the potential collaborations with IMPC. What we need to do is develop and establish a more extensive behavioral pipeline, maybe taking advantage of what was done in IMPC. Especially behavioral testing in combination with imaging, like the multiphoton microscopy. A generation of new mouse models is also important. We used to collaborate with Munich, but I think we have to expand that. And of course, our researchers, and I really urge them to make the connection between IMPC and the IMPC models and the IMPC phenotyping data and candidate genes they identified either in clinical studies or other mechanistic studies. And that urge them really to use IMPC mouse models and the phenotyping data to find out if there is already a model available. And regarding the home cage monitoring, I'm not sure if that someone already mentioned this during the conference, but maybe some of you know, and Sabine Hölter and Silvia Manilo, they're also very active in this cost action, and we had a couple of webinars even together. Cost action tea time, that is about improving biomedical research by automated behavior monitoring in the animal home cage. I think together with what I see at the JAX, for example, in the United States, development, a really good development that we need to kind of make use of the 24/7 data collection of behavioral data in a home cage and use that for phenotyping. And this cost action is there to develop a kind of roadmap how that could be done. And there are many activities, and I think this is really a very important and very good occasion to team up here, both my institution, IMPC, but also the cost action. Okay, and the last but not least platform is the bioinformatics platform. All this is classical, but I think as it appears at the end, it's kind of unique as we have it here in Luxembourg. The bioinformatics platform, or the core facility, provides a centralized resource of expertise in computational biology and statistics. And we offer support as the other platforms, so at all stages of a research project. And this is also about storing the data. It's about GDPR, which is really an issue here in Europe. It's about curation of existing clinical data. It's about introducing and maintaining international standards, for example, FAIR principles and principles regarding quality control. But we also have things like pre-registration of studies, what not everybody is doing, and also something that's called a pre-publication check. That means when you want to submit a paper to a journal, we have an internal kind of quality control process where we really check, are all animal and cell models, is the nomenclature, for example, correct? Are all chemicals and that were used correctly cited? Is the data stored somewhere where it's accessible and following the FAIR principles, etc., etc.? And unless and this pre-publication check is really mandatory for everybody who is publishing or submitting a paper as first or last author. And then all this, of course, requires also extensive training of researchers by the platform. We're currently restructuring the platform because there have been some personal changes, but still there is one thing, and I guess some of you are waiting for this. We still have the clinical blood chemistry paper in the pipeline, and the plan was, and announced this earlier this year, that we could team up with experts here from LCSB, for example, in machine learning to include a state-of-the-art analysis of this data set. And this is ongoing. I still don't have results I could show, but the key messages for that paper, I guess you would remember that, is, of course, first of all, the blood chemistry profile of Black 6N mice, then the sexual dimorphism, and I also think a very important message of this paper is that batch measurements, like the different centers in IMPC that contributed data or to this whole data set, that this increases robustness. I think it's a really important point, and it's coming up everywhere, and people need evidence. So examples where this worked, and of course the link to human data. And I already frequently get questions from people like Chris or Birgit Radkolb or others, what is happening, but I will reach out to them when I have first results from our people here, and then hopefully finalize and submit the paper this year. Good, so the last slide is, I have to move that. I'm really sorry, this is so confusing here. So I hope I could show you that the platforms here, and I think this is really a general thing, are not only offering highly qualified service and transparent project management and communication between clients and researchers on the platform, but it's also about consultancy. It's about training of users. It's about technologies, actually. So the maintenance and also the development of new methods here in the R&D sector is really important to make sure that there is these platforms or research infrastructures have a future, because we cannot assume that research infrastructure has never exists forever. But if there is no use, no request, and if the quality of the project is going down, it's also getting to an end of the pipeline. This is, of course, what we do not want. And so the last sentence here, the rationale of the research infrastructure is to offer scientific excellence, to continuously adapt to the needs of the research community, and to identify synergies. And with that, I want to close. I want to thank all platform managers here in Luxembourg and all scientific and technical staff. They made it actually very easy for me here to start the new job. The support teams, the PIs, and the director, Michael Hennecke. And I thank you for your attention, and apologize again for this a bit chaotic presentation.
Thank you so much,
questions?
Jan. I think you did it well. You were on time. Maybe we have a chance for 1 or 2 questions. Not yet. Okay, so you have a community of users which seems to be very close to your platforms. So do you have any let's say user committee which are in place to, or how can you manage to put the direction on the scientific platform? So what should be developed in the close future? Is it coming from the users, from you, from both?
From both. This is a good question. We have a kind of governance for every platform, and the platform managers, they have their user committees, and there is actually a close exchange between these people. Whenever we introduce something new, like project management software or new technologies, it's discussed with the user committee. They're meeting once a year at least, and single users. Then I have or invite people for external evaluation. So this is my way of looking at the performance of the platform. But innovation is coming in this process. Users, but not only PIs here from the LCSB, but also from university, from other institutions in Luxembourg and beyond. So this is a very open and transparent process.
Very good. Yes, Anne?
Okay, so Jan, so do you also keep archives of the data or the samples? And do you have accredited SOP processes?
That's a good question. You can imagine that all the imaging data or the data that has very high volume, the storage is cost intensive. Let's say, think about the end of the project. If you submit a paper to a journal, they require a proof that the data is stored somewhere, and we offer that. So especially the bioinformatics platform is offering storage place. For other platforms like the sequencing, we are handling human data, and due to GDPR, we only have the data for a limited time, and then it's cancelled. So we can't store these data. Personally, I would think this is not optimal, but there is, from the legal perspective, no other way. So GDPR is really an issue there, and regarding the other platforms, it's a case-by-case thing.
Okay, last question, Rose.
Good morning, Jan. Just a question. You mentioned that you have a user committee. How did you constitute the committee? How did you decide who gets to be on the user committee? Thanks.
So the user committee is really every user that is currently using the platform or planning to use it or has used it. So I wouldn't say it's who has ever used the platform, but everybody who's interested to join is very welcome. So we really need this input from people. We even thought about having outside people there in this consultancy process when it's about, for example, purchasing a new machine. but in general, it's all the current users and those who are planning to use it or just have used the platform.
Okay. Thank you so much.